Let's think of the process of synthesizing a protein in terms of building a bracelet. Here is a completed bracelet that I want my bracelet-making robot to build. Let's review how this process works. Here's our DNA coding strand. Let's imagine that this DNA strand holds all of the instructions that my robot will read and utilize to eventually make my bracelet. I give the DNA strand to my robot and he makes a copy of the instructions. However, the instructions encoded in DNA are not in a language that my robot can understand. Therefore, before we can start building the bracelet, or our protein, my robot must first transcribe our DNA code into an mRNA code. Now my robot can read the mRNA instructions. Each group of 3 nucleotide bases, or instruction letters, codes for a special bracelet bead, or amino acid, that will be added to our bracelet. Our robot knows to start bracelet building when he sees the start codon, AUG. Reading the mRNA strand from this point, 3 letters at a time, our robot finds the associated bracelet bead or amino acid for each code. Here is our resulting bracelet. Now, in a perfect world, my robot would make the correct bracelet perfectly every time. However, not even robots are perfect. Sometimes my robot copies down the DNA instructions incorrectly, switching up some of the letters. This results in point mutations, a type of mutation where one base or instructional letter is swapped for another at a specific location. Point mutations can have a variety of consequences on our final bracelet product. Let's look at a few scenarios that have occurred due to my robot messing up. Here we see that my robot copied down a G instead of a C. The C to G switch up is called a transversion. Transversions occur when a pyrimidine base is swapped with a purine base or vice versa. This change in our DNA will trickle down and create a change in our mRNA transcript. However, when our robot translates our mRNA instructions into the bracelet sequence, we see that the same bracelet bead or amino acid results. This is because multiple different codons can code for the same bracelet bead or amino acid. This example is a silent mutation, which is a point mutation that results in an unchanged polypeptide sequence. Even though my robot copied the instructions wrong, we still got the correct bracelet or protein product in the end. In this example, our robot copied down a G instead of an A. The G to A switch is a transition. Transitions occur when a purine base is replaced with another purine base, or a pyrimidine base is replaced with another pyrimidine base. Our mRNA transcript is changed because of the change in DNA, but this time our final bracelet pattern or polypeptide sequence changes as well. This new 3-letter codon encodes for the amino acid arginine or a blue bead instead of lysine or a pink bead like we want. What we are seeing here is called a missense mutation, a point mutation that causes a change from the bracelet bead or amino acid originally encoded. Now the bracelet pattern or polypeptide sequence no longer matches the one that we wanted. Here, my robot switched an A with a T, a transversion. This mistake once again changes the mRNA transcript, but in a new way. We can see here that this codon, which previously encoded for lysine or a pink bead, will now code for the robot to stop bracelet making early. This is known as a nonsense mutation and means that our bracelet or protein will be incomplete and not contain enough beads or amino acids. Finally, I have seen my robot cause read-through mutations. Here, another transversion occurs where an A is replaced with a T. With this change, the original location of the stop codon now encodes for another bracelet bead or amino acid. This makes our bracelet longer than we wanted, and now my robot has no idea where to stop the bracelet. He keeps reading the instructions and adding new bracelet beads that we did not want on our original bracelet.